Welcome back, Biology 205 students, to part 2 of the introductory lecture on an introduction to microbiology. What we're going to do in this part of the lecture is, as promised, we're going to take a little tour of the world of microbes and learn one or two examples of all of the different types of microscopic organisms there are. Remember that we have bacteria, we have viruses, we have fungi, we have yeast, we have helminths, and we have protozoa or protists, which I'll explain the difference between a protist and a protozoa later. And so we'll learn one or two examples of each, and these one or two examples could easily be on quizzes in the near future. So we'll ask a few practice questions afterwards to see how well you've learned about these organisms. So let's take a little tour. All right, starting with this list on the right-hand side of the slide. Humans, the genus name and species name, genus Homo, species sapiens. Humans are, of course, multicellular organisms, and they're composed of billions of individual eukaryotic cells. So humans are members of the domain eukarya. Humans are eukaryotes. The body is organized into different tissues, and those different tissues form different organs, and the different organs carry out different functions in the body. So you're aware, of course, that the kidneys, for instance, their job is to filter urea out of the bloodstream and get rid of it in the form of urine. The lungs' job is to bring in oxygen and remove carbon dioxide that's used for cellular respiration. And so the body of a multicellular eukaryote is generally organized into what is known as organ systems. All of the cells that make up the human body are, of course, eukaryotic cells because they are all large. They all have a nucleus. They all have linear chromosomes. Each cell in the human body contains 2 copies of every gene that humans are supposed to have. So every gene that humans are supposed to have is referred to as the human genome. Every cell in the human body has 2 copies of the human genome, and whenever you have 2 copies of the genome, you are said to be diploid. Right? So humans are diploid organisms. We'll see later on that most of the microbes that we deal with are haploid, meaning that they have only 1 copy of the complete genome in the cell. But humans are diploid. We have a complete genome that's made of about 23,000 different genes, and those 23,000 different genes are distributed among 23 chromosomes. The abbreviation for chromosome is exosome, as you see there. And we have 2 copies of each one of those chromosomes. We generally number them chromosome 1, so we have 2 copies of chromosome 1. We number them chromosome 1, chromosome 2, chromosome 3, and so on. So we have 2 of each of those chromosomes. The cells that we are made up of do not have a cell wall. Now, there's sometimes a bit of confusion amongst biology students. What is the difference between the cell wall and the cell plasma membrane? Well, the cell plasma membrane, if you remember from first year biology, the cell plasma membrane is made out of phospholipids, whereas the cell wall is something that you learned is something that plants have, for instance. So all plant cells are surrounded by a polyhedral wall that's made of cellulose and sometimes lignose. And bacteria also have a cell wall. Bacteria are some of the interesting organisms on Earth that have a cell wall, but their cell wall is not made of cellulose. It's made of a different material, a polysaccharide called peptidoglycan. So we'll learn all about peptidoglycan when we get to the bacteria. But for now, the 2 main types of organisms that have a cell wall are the plants and the bacteria. The 2 cell walls that they have, the types of cell walls are made out of different materials. Plant cell walls are made out of the polysaccharide cellulose. Bacterial cell walls are made out of the polysaccharide peptidoglycan. Human cells and most other animals do not have cell walls. But all cells, whether they're a plant cell or a bacterial cell, the cell itself is bounded by a flexible membrane that's made of phospholipids that's called the plasma membrane, the cell plasma membrane. And of course, the main difference between a eukaryote and a prokaryote is that eukaryotes have a nucleus. The eukaryotic cells contain their genes inside a structure called a nucleus. Humans, by the way, have linear chromosomes, which mean the chromosomes look like a little X that you've seen, right? They only look like that when the cell is dividing. They don't look like that otherwise. But nevertheless, the human chromosome is a single linear chromosome as opposed to a bacterial chromosome, for instance, which is a circle, where the DNA is formed into a circle. Okay, so here are the main characteristics of eukaryotic cells. Eukaryotic cells contain their genomes inside the nucleus. They're fairly large and they're bounded on the outside by a plasma membrane. The plasma membrane is made of phospholipids. They contain smaller organelles, like we just mentioned, the fact that the human body is divided up into organs that carry out specific functions. Well, eukaryotic cells are made of organelles. They contain smaller structures that carry out different functions that we call organelles. And some of those organelles include mitochondria. Mitochondria, their job is to produce extra energy for the cell, produce it in the form of ATP. We have the rough endoplasmic reticulum, which is where proteins that are intended to be exported outside of the cell are translated. We have the smooth endoplasmic reticulum, which is where lipids are created. We have the Golgi complex, where proteins that are meant to go outside the cell, proteins that are created for purposes of export, are glycosylated. And in plant cells, which are also considered to be eukaryotic cells, plant cells additionally have chloroplasts, which are responsible for photosynthesis. All right, so eukaryotic cells have organelles, including a nucleus, mitochondria, that's the plural, mitochondrion is the singular. They have a plasma membrane, or known as the cell membrane, which surrounds them on the outside. They, if we exclude plants, they do not have a cell wall. So if we're just talking about animals and fungi and things like that, they don't have a cell wall. They just have a plasma membrane. They tend to have linear chromosomes. Most eukaryotes have linear chromosomes. Most eukaryotes are diploid. They have 2 copies of each gene, therefore 2 complete copies of the entire genome in every cell. Their genome is made of DNA. Now that's an interesting point, because have you ever heard of an organism that has a genome that's made of anything else? Well, in this course we're going to learn that there is one thing on Earth that has sometimes has a genome that's made out of RNA, and those are the viruses. There are generally 2 broad types of viruses. One type of viruses has a DNA genome, and another type of virus has an RNA genome. Colds are caused by a virus called rhinovirus, for instance. Rhinovirus is a virus that has an RNA genome. You will not find any DNA inside a rhinovirus. The COVID-19 disease is caused by the coronavirus, caused by the coronavirus, and coronaviruses are another type of virus that have RNA genomes. So again, you will not find any DNA inside a coronavirus. Generally, what the organisms that have a DNA genome do is they make an RNA copy from a gene, call that a messenger RNA, that is created inside the nucleus by RNA polymerase enzymes, and then transported out into the cytoplasm, where it is translated into protein by ribosomes. And as I said earlier, if the protein is meant to be exported out of the cell, the protein is translated inside the rough endoplasmic reticulum, where it is created, and then it's transferred from the rough endoplasmic reticulum to the Golgi complex, where it is glycosylated and then sent out of the cell. All right, just to give you an idea of the size difference between a prokaryote and a eukaryote, I mentioned the fact that eukaryotes tend to be quite a bit bigger than prokaryotes. So in this picture, we see a macrophage. A macrophage is a white blood cell that you would find in the human immune system or any other animal immune system. And their job is to eat bacteria and clean the blood and to clean the body of bacteria. So here we see this gigantic macrophage, and it is eating these little tiny bacteria here. So there's a bacteria. Right, there's a bacteria. Here's a bacteria on the surface of the macrophage. Right, there's another bacteria. So you can see the size difference. So this brings up an interesting point, which is that there are white blood cells in the human body whose job it is to eat bacteria. Their job is to destroy bacteria that are invading the human body by eating them. Right? Now, these white blood cells are pre-programmed to eat these bacteria. They don't have to learn how to do it. they automatically start eating certain types of bacteria as soon as they see them. And so they have an innate ability to destroy specific types of bacteria. For instance, we're going to learn about a type of bacteria called Staphylococcus aureus, Staph aureus. And Staphylococcus aureus is one of the types of bacteria that the macrophages are automatically programmed to start destroying as soon as they see them, the second that they see them. And so whenever you have a bacteria get into the body that these white blood cells are pre-programmed to destroy, these white blood cells will swarm to the area and start destroying it. And they will eat a lot of these bacteria. Eating too many of them will cause them to die, so they will die. But they'll all swarm to this area, and they will create pus. We call pus. Right, so pus is that kind of sticky, smelly substance that you get on a cut if it's infected. That is pus. And what that pus is made of is billions of dead macrophages, millions, billions of dead white blood cells that are there to kill a bacteria that is infecting that particular wound. Right? So that's what pus is, and any bacteria that provokes the formation of pus, that automatically triggers these cells to come and start eating them, any bacteria that does that is referred to as a pathogenic bacteria. The formation of pus by specific bacteria is referred to as the pathogenic response, and I'll show you how to spell that later. And the pathogenic response is triggered by bacteria that we classify as pathogenic bacteria. So when we get to the section on bacteria for the course, we'll talk a fair amount about different types of bacteria that are classified as pathogenic bacteria. All right, what about helminths? These are the microscopic worms. Okay, this is an example of a helminth called Diphyllobothrium latum. Diphyllobothrium latum is a helminth that invades, it's an example of something called a tapeworm that invades fish, and it gets into the tissues of fish and lays eggs inside the fish muscle tissue. And then when we eat that contaminated fish, these eggs hatch, and then they get into our intestine. And these little microscopic worms grab onto the inside of our intestine. They latch onto the inside of our intestine and start absorbing our food. So as our food is traveling through the small intestine, these types of organisms will absorb it, absorb a lot of it, and will get bigger and bigger. They will grow the more food they absorb until they become enormous. So they start out being microscopic and therefore qualifying as microscopic organisms. But if they stay long enough in your intestine, they will become enormous, up to 10 meters in length. So you are in danger of getting diphyllobothriasis, which is the disease caused by Diphyllobothrium latum. You're in danger of getting this disease if you eat raw fish or if you eat undercooked fish. So this is a hazard of people who like to eat sushi, for instance. The Japanese food sushi is a delicacy where you eat raw fish. I happen to love it. I was born and raised in Vancouver, and we have a lot of sushi restaurants in Vancouver. So I love my sushi. I can't eat enough of it. I would eat it all day if I could. But I am running the risk of contracting Diphyllobothrium latum if the fish is not properly prepared. In Japan, the way that you're supposed to properly prepare the fish is you're supposed to flash freeze it. So you freeze it in liquid nitrogen or something like that, and you quickly thaw it out, and hopefully it wasn't frozen long enough that it destroyed the texture of the raw fish, but hopefully it was frozen long enough that it killed the eggs of any tapeworms that are in there, but it wasn't frozen long enough to destroy the texture of the fish. So there's a very special technique to doing that. That's what they're supposed to do in Japan, but that's not what they do everywhere else in the world necessarily. So they're supposed to be doing that in Vancouver, but you will occasionally find sushi restaurants where they don't realize that. And if you ask them where they get their fish, they say, well, we buy it directly from the fishermen. Down in Vancouver, we have Fisherman's Wharf down by Granville Island, where you can buy fish directly from the fishermen off the dock. And it's a bad idea to buy that fish and eat it without cooking it. So it's all right to buy fish from the fishermen, but you just have to make sure that you cook it very, very well. Otherwise, you might get Diphyllobothrium latum. Or you can freeze it quickly in liquid nitrogen, like they do with sushi, the proper way to prepare it. But not many people have the facilities for that. So if you're just an amateur person making your own fish, you should always cook it very well to avoid tapeworms. All right, so the helminths are microscopic worms. They are multicellular, so they're made of several, generally they start out being made of several hundred cells, and then they get bigger and bigger. They're made of more and more cells. The cells that the helminths are made up of are eukaryotic cells. So helminths are classified as eukaryotes. They are a human parasite as well as a fish parasite. They invade and colonize human tissues as well as fish tissues. Obviously, they don't have a cell wall. They don't have a cell wall that's made out of either cellulose or peptidoglycan. They have a DNA genome and linear chromosomes. I probably should have warned you before showing you this picture, but this is the inside of a human colon, the inside of a human large intestine, where you see the Diphyllobothrium latum having grown to be quite large. And it looks kind of like ticker tape. I don't know if you've ever seen that paper tape that's kind of ruffled at the edge that people put around the room when they have birthday parties and celebrations. And that's where they get the name tapeworm, because they look like a tape, ticker tape that you put out. And so if you eat one of these, if you ingest one of these eggs in undercooked fish, it will hatch inside your intestine. It will grab on to one end and by the head, and then the tail of the tapeworm will just continue to grow until it's 10 meters long, up to 10 meters long. At that point, you'll become very sick. You'll get jaundice. You'll turn yellow. Your skin will turn yellow in color. You will lose a lot of weight because the tapeworm is stealing most of your food that you eat. And then they'll have to find some way to kill it. If it's this big, usually they have to remove it surgically. They have to cut you open and then pull it out. But that's an extreme case. Okay, so the disease that's caused by Diphyllobothrium latum is called Diphyllobothriasis. Diphyllobothriasis. There's another worm that is it's another type of a worm called a nematode worm called Trichinella spiralis. And Trichinella spiralis you can catch if you eat meat, wild meat that you have hunted. So if you go out and you shoot a deer, or you shoot an elk or a caribou or something like that, which is something that people do in British Columbia, you eat that meat without having cooked it thoroughly, you run the risk of contracting a disease called trichomoniasis. Trichinosis, disease called trichinosis, which is these microscopic worms get into your muscle tissue and start eating away at your muscle tissue. Now, this is an extreme case of trichinosis, but you always run the possibility of catching trichinosis if you eat wild animals that you've hunted in the forest. And there are a lot of people in British Columbia who do that. For instance, I had an uncle. He's no longer with us, but he used to he lived in Quesnel, and every fall he would go out and shoot a moose and then cut up the moose and put it in the freezer, and then the family would eat. My cousins and my aunt and uncle would eat moose meat all winter, and sometimes they would bring some of the moose meat to cook when they visited us in Vancouver. And they would make moose meatballs and moose hamburgers and things like that. And I didn't like the taste of it very much because it tasted kind of bland compared to regular beef. But nevertheless, they considered it kind of to be kind of a delicacy. But now I realize that was a possible way to catch trichinosis. Okay, so two examples of helminths: Diphyllobothrium latum and Trichinella spiralis. Let's look at some protozoa. So protozoa are single-celled eukaryotes, and they have a very diverse morphology. There are a lot of different types of them that are very strange by our standards. They are constructed in a very strange way by human standards and by the standards of modern organisms. And the reason for that is because the protozoa, the protists, evolved before nature had really decided which life form that it would favor first, right? So the protists evolved during a period of life on Earth when basically nature was trying a lot of different things to see what would work best. That's a good way to put it. They were trying a lot of different things to see what would work best. And so we have all kinds of bizarre things that we can now consider to be bizarre, but they were just trying new things at the time. So this is Giardia lamblia. Notice that it has two nuclei, right? These things that look like eyes, it actually has two different nuclei. And that's some of the protists have two nuclei. Some of them have two nuclei. One of them is big and one of them is small. And the big one is called a macronucleus and the small one is called a micronucleus. Some of them are made, these are diatoms, sorry, these things over here are diatoms, which are a form of protist, which is very common in the ocean, and they have a body that's made of silicon. Now you know that most life forms on Earth today are made out of carbon, hydrogen, and oxygen. Those are the carbon-based life forms. But before the carbon-based life forms caught on, before they became a trend, some of these protists evolved which had a silicon body. So you know that on the periodic table, silicon falls right below carbon. Silicon is capable of forming 4 bonds, and so is carbon. So we went through a period of life on Earth about 2 billion years ago where nature was trying to decide which one is best to build living things out of. Is it best to have living things that are made out of carbon or carbon compounds, or is it better to have things that are made out of silicon compounds? They both have good things, they both have similar characteristics. They both silicon and carbon form 4 bonds under the right circumstances. So you can create a lot of diverse macromolecules, right? So there are a lot of protists that are still around that have a silicon body, which is unusual by our standards, but that's a thing. These are trypanosomes. This is a protist that infects your blood. It's carried by insects, and it can give you a disease called sleeping sickness. Those are quite unusual looking. And then these things here are called dinoflagellates, which are another ocean protist that creates highly poisonous toxins called red tides. But all of these things are quite unusual looking and quite built in a bizarre way by our standards. And they're like that because, as I said, they were formed during a period in evolution on Earth. They were formed during a period where nature was trying a lot of different things just to see what worked best. Eventually, some things ended up working better than others, and they evolved. They continued to evolve, whereas other things didn't work quite as well, but they're still around. Those old things that didn't work quite as well are still around, and in fairly large numbers in some places. So just to give you another example that I like to use, most of you are too young to remember this, but I remember when we had videotape. This is in the 1980s, and there were 2 companies. There was VHS and there was Betamax. These were 2 different companies that made videotapes, and they both were good. They were both acceptable types of videotaping. There was no good advantage to one over the other, but they were in contest. They were in competition with one another, and eventually VHS won. So pretty soon, 90% of the people that had videotape machines had VHS recorders, but there were still 10 or 15 or 20% of the people still had Betamax tape recorders, and they were still around. Right? So the same thing is true about many of these protists. The best ones became more abundant, but the old-fashioned ones that didn't really catch on as well, they're still around. And so that's what we have in protists. So the same thing is just looking at all of these different sound recording devices. Right, so here we have, this is a reel-to-reel tape recorder, which was very popular when I was young. The reel-to-reel tape recorder gave way to the cassette tape recorder. Way back in the 1920s, we had gramophones to record sound. Now we have CDs, and then eventually CDs gave way to MPEG-3s and then MPEG-4s and things like that. So just because we eventually evolved a very sophisticated technology of having MPEG-3s and MPEG-4s doesn't mean that there are not still people around who have cassette tape recorders. And so the same thing is true of protists. There are many protists around, and they are unusual by modern standards, by the standards of modern organisms, and they reproduce in strange ways, and they're constructed in strange ways. With some of them have more than one nucleus, and some of the nuclei do different things. But these are all things that nature was kind of trying out about 2 billion years ago, and some of them are still around. All right, now here is one of those protists called Plasmodium vivax. Ironically, Plasmodium vivax is very closely related to algae in the ocean, but it happens to be carried by animals on land as well. And so what happens is that Plasmodium vivax is a protist that causes disease called malaria, and malaria is carried by mosquitoes. It's carried between humans by mosquitoes, and it's carried between humans and monkeys by mosquitoes. There are several different several different species of Plasmodium that all belong to the genus Plasmodium. There's Plasmodium vivax, Plasmodium falciparum, a few other ones, and they all cause malaria. So an example of a protist is Plasmodium vivax, which causes malaria. Notice that the Plasmodium vivax has a single nucleus, right? So this is a single cell. It's not a multicellular organism like a helminth. It's a single cell that is kind of worm-shaped in this particular form. And if you stretch it out, it's about 10 microns. It's about 10 microns long and only probably 0.1 micron wide in this particular life cycle. It has several different forms that it exists in depending on which form that you find it. But this is one of the forms that Plasmodium vivax takes on as it's developing. And so Plasmodium vivax causes malaria. Plasmodium vivax is an example of a protist. Okay, so Plasmodium vivax, the protozoa are single-celled eukaryotes. They have one, or in some cases more than one nuclei. They generally have a DNA genome, and I don't know any that have an RNA genome, in fact, so they do have a genome. But some of them, a DNA genome, but some of them are haploid, and while others are diploid. It depends on the organism, depends on the species. They tend to be mobile, or motile is another word for the ability to move around. Some of them are parasites, and some of them have a cell wall. In fact, as I mentioned earlier, some of them even have a cell wall that's made out of silicon. Some of them have a cell wall that's made out of silicon. Some of them have a cell wall that's made out of cellulose, amazingly, because, not really amazingly, because we know that the plants evolved from some protists. There's a family of protists that has a cellulose cell wall, and that family gave rise to that family evolved into the plants. But the ancestors of the plants, the protists that evolved into the plants, those ancestors are still around as well. Okay, malaria is caused by Plasmodium vivax, as I mentioned, and it is carried by mosquitoes. And we'll learn later on that when we're talking about diseases that are transmitted between people or between people and animals by an insect, we call the insect a vector. Right? So in this case, Plasmodium vivax is transmitted from person to person by a mosquito vector. A mosquito vector. So we're going to learn all about different types of vectors for different types of diseases as the course goes on. Okay, these are Giardia lamblia. We have these in British Columbia. They are endemic to British Columbia as well as other places in North America. They are kind of unusual. They have two nuclei. Right, these things, they kind of look like owls, or they kind of look like owls that are staring at you from a tree. And these two eyes are actually two different nuclei. So this is an unusual thing that nature was trying out at one point. Is it necessary to have two? Is two better than one? I don't know. They didn't, we continued to evolve with cells that have only one nucleus, and so we're okay. And notice that they're swimming around with these things. These are flagella. Right, so Giardia lamblia lives in the water. It usually gets into the water from excrement, usually animal excrement, specifically usually from beaver excrement. Right, so if you're hiking in the woods and you come across a lake where there are beavers, don't drink the water from that lake, or you may get giardiasis. Okay, so they're using flagella to swim. They cause a disease called giardiasis, which is a form of diarrhea. All right, here's another important protist. This one is called Toxoplasma gondii. Toxoplasma gondii also has two nuclei. Right, so you can see the two nuclei in this picture here. And they're about to divide here and so on. Now the interesting thing about Toxoplasma gondii is that it is something called it is also classified as a teratogen. Teratogen, the word teratos is an ancient Greek word meaning monster, and the word gen means to generate or to cause something. So teratogen is a word that means something that causes monsters to appear, and this is a very politically correct term, but what it means is teratogen is specifically used to refer to things that cause birth defects in unborn children. Right, so you know that there are things that can cause mutations or they can cause problems in children as they're developing in the mother's womb, and any of those things are classified as teratogens. Right, so Toxoplasma gondii is classified as a protist. It's classified as a eukaryote. It falls into many different classification schemes. But one of the things that we also apply, one of the terms that we also apply to Toxoplasma gondii is that we say that it is a teratogen. Right, so a teratogen, if you get infected with if you're a pregnant woman and you get infected with Toxoplasma gondii, your child may have birth defects. So you always have to avoid contracting Toxoplasma gondii when you're pregnant. In fact, for adults, an adult who gets infected with Toxoplasma gondii usually doesn't even know it because it is a subclinical infection in adults. The term subclinical means that it doesn't have any symptoms. You don't feel any effects from having this disease. So many adults contract toxoplasmosis, which is what the disease is called. Many adults contract Toxoplasma gondii and get toxoplasmosis, but they never know it. Unfortunately, however, if a pregnant woman contracts toxoplasmosis, the baby could have a variety of different birth defects when it's born. So anything that causes birth defects if it infects a pregnant woman is called a teratogen. Toxoplasma gondii, unfortunately, is carried by domestic house cats, and in particular, you can catch it quite easily from cat feces. Right, so a pregnant woman should not be cleaning out the cat litter box. She should get somebody else to do that, for instance. So if you're pregnant, avoid, especially in the first month, the first 1 or 2 months during the period of organogenesis, when the organs are forming, you should avoid playing with cats or cleaning out the cat litter box if you're pregnant, especially during the first trimester. The first trimester, of course, is the first 3 months of pregnancy. Okay, so the 3 examples of protists that we talked about are Plasmodium vivax, Giardia lamblia, and Toxoplasma gondii. Let's talk about yeasts. Okay, here's an example of a yeast. You already heard about it in the other lecture. This is Saccharomyces cerevisiae. Saccharomyces cerevisiae is one of the main microbes that is used to do fermentation. So fermenting grape juice into wine, fermenting barley into beer, fermenting rice into sake, fermenting other alcoholic beverages is usually carried out by Saccharomyces cerevisiae. It's also, of course, it's also used to make bread. So if you mix flour and water and sugar together, and then you put Saccharomyces cerevisiae in it, the Saccharomyces cerevisiae will start to eat the sugar. It'll eat the glucose, and then as a waste product, it will produce carbon dioxide. That carbon dioxide will form bubbles in the bread, which cause the dough to rise. Right, so Saccharomyces cerevisiae is sometimes colloquially referred to or commonly referred to as baker's yeast or brewer's yeast. Right, so whenever you hear the term baker's yeast or brewer's yeast, we're referring to Saccharomyces cerevisiae. And I might ask you that on a test. Right? So I might ask you, when I say baker's yeast, what am I talking about? And the answer is Saccharomyces cerevisiae. Yeast cells are all around us, and in fact there are billions of yeast cells over every square centimeter of our skin. Right? So if we touch things with our hands, if we touch things that are meant to be sterile, there's a very good possibility that we can contaminate them with Saccharomyces cerevisiae on our skin, or another common yeast, which I'm going to teach you about, called Candida albicans. So every square centimeter of human skin and the skin of any other animal and every square centimeter of a tree or a plant or anything like that is generally covered by millions, if not billions, of yeast cells and bacterial cells. And two of the most common yeast cells that cover everything are Saccharomyces cerevisiae and Candida albicans. Okay, now Saccharomyces cerevisiae is classified as something called a budding yeast. Those of you that have taken Biology 200 from me know the difference. So you notice that these yeast cells, they're shaped kind of like bowling pins. They have a large circle and a small circle. The small circle is called a bud. And what that means is that the cell is dividing in half. These cells are in the process of dividing in half, but they do not divide symmetrically. The two daughter cells that result from the division are not the same size. A big cell divides into a big cell and a little cell, and then the little cell will grow until it's big. Right? So that is referred to as budding rather than fission. Fission means something splits in half and then it splits into two equal halves. So there are some yeasts that reproduce through budding, and there's some yeasts that reproduce through fission. Right? So Saccharomyces cerevisiae is classified as a budding yeast. There are other yeasts that are referred to as fission yeasts. Okay, so yeasts are classified as single-celled eukaryotes. They are some of the they have one nucleus. They're a little bit small compared to regular eukaryotic cells like human cells, for instance. So protists tend to be the largest eukaryotes. Those protozoa can be 50 to 100 or even 200 microns in length. Human cells tend to be smaller. They're around 7 or 8 microns in diameter. Yeast cells are a little bit smaller than human cells, and so you can recognize them under the microscope because they're a little bit smaller. They tend to be around 5 microns or something like that. They have a DNA genome. There are no yeasts that have an RNA genome. They have linear chromosomes, not circular chromosomes, and they do have a cell wall, and that cell wall is made of chitin, a polysaccharide called chitin. Chitin is very closely related to cellulose. There's only one small substitution. I don't expect you to know the difference in structure, but I do expect you to know that plant cells have a cell wall that's made out of cellulose. What do bacterial cells have a cell wall that's made out of? Which polysaccharide makes up bacterial cells? Do you remember? Peptidoglycan. Okay, then yeast and fungi both have cell walls that are made of a polysaccharide called chitin, C-H-I-T-I-N. By the way, in biology, every time you see the letter CH, you usually pronounce it as a K, not a ch. And that's because many of the words in biology came from Latin, and the Romans spoke Latin, and they did not have a soft C. They never pronounced the C as S or sh. They pronounced it as ka, usually ka. So in the original Latin, for instance, you know who Julius Caesar was. Julius Caesar in the Romans would never pronounce his name like that. The Romans would pronounce his name Iulius Caesar. And then it was only until the Europeans got a hold of the Latin language and changed it and degraded it basically, that we started adding soft C's and ch sounds. So the biologists, in the early days of biology, most of the biologists knew how to speak proper Latin, and so when they named these things, they would pronounce it as a k, not a ch. We've been kind of fighting a losing battle since then, and some words that begin with ch have snuck into biology that we pronounce as a ch, but chitin is not one of them. So you pronounce this word as chitin. Okay, so yeast and fungi are two organisms that have every cell is surrounded by a wall that's made of chitin. Okay, now if you think back to Biology 110, can you think of any animals that have chitin in them? Can you think of any animals? Right. All right, if you can't think of any, I'll tell you the answer. The arthropods. The arthropods have an exoskeleton that is made of chitin. Right. So an insect has an exoskeleton, and that exoskeleton is made of chitin. The crustaceans, like the lobsters and crabs and so on, have an exoskeleton that's made of chitin. and the arachnids like spiders and ticks and things like that have an exoskeleton that's made out of chitin. Dust mites that eat dead skin in your mattress, those have an exoskeleton that's made of chitin. So what do yeast cells have in common with mosquitoes? They both have an outer coating that's made of chitin. Now, one of the interesting things about chitin is that it's highly allergenic. Allergenic means that it'll give you an allergy, it'll make you sneeze, right? So that's why many people are allergic to yeast and fungus. Fungus will actually cause, if you inhale fungal spores, it will usually cause a really bad immune reaction, a hypersensitivity reaction as it's called, and cause your body to go into what's called anaphylaxis. Anaphylaxis means that the fluid leaks out of the capillaries and you get swelling and a drop in blood pressure. So inhaling fungus is not a good thing. It causes the body to have hypersensitivity reactions because chitin is a very allergenic polysaccharide. It causes you to have allergenic reactions. When you sneeze, because there's a lot of dust in the house, you're usually sneezing because you're inhaling the skeletons of dead dust mites that have been eating the dust. The dust mites are not eating dust per se, they're eating dead skin, dandruff and so on that you usually find in mattresses and so on. So that's why a lot of things will make you sneeze, but it's usually because of the chitin that you find in either fungal molds or dead insects. If you go into a library where there are a lot of books, the library climate is controlled very carefully to make sure that the humidity is low, because if the humidity is high, fungus will grow on the books. And fungi are allergenic, so they make your eyes swell up and they make your nose run and you make your eyes feel itchy if you have fungus growing on the books. Fungus will, because most fungi produce cellulase enzymes, they are actually able to digest or break down cellulose. And where do you find cellulose? You find cellulose in paper, right? So left to its own devices, a lot of fungus will grow on books if the books are allowed to get wet. Right, so if you go into a well-organized library, you will not have itchy eyes and a runny nose because the humidity in the library is controlled. If you go into a used bookstore where they don't control the climate, you'll notice that many times you kind of feel sick when you go into a used bookstore because these books have been damp. You know, people have been leaving them outside and then donating them to the bookstore or whatever, and then they grow mold, they grow fungus on the books. So that fungus is allergenic and it causes an allergy reaction. Okay, so yeast and fungi both have an exoskeleton made of chitin, and the chitin exoskeleton is allergenic, which can cause various problems. Notice that fungi and yeast are, the difference between fungus and yeast is very simple. Fungus can exist in multicellular colonies, and yeast only exist as individual cells, right? So that's the very quick, a very quick and simple definition that allows us to tell the difference between fungus and yeast. Otherwise, they're the same. Basically, they're very similar, except for the fact that fungi can exist as a multicellular colony, and yeast only exists as a bunch of single cells. Right? Okay, now both fungi and yeast can reproduce sexually, and we'll explain how that works later. But they can also reproduce vegetatively. Right, so yeast can reproduce by sexual reproduction, or they can reproduce through binary fission or budding, which is considered to be vegetative reproduction. And furthermore, most of the yeast are not motile. Most of them are vegetative, meaning they just sit in the same place. Vegetative state means you sit in the same place and don't move. Okay, so vegetative in this case means that they don't move and they reproduce through fission usually. All right, here's another type of yeast which you need to know all about. This is a yeast that's commonly found on the skin along with Saccharomyces cerevisiae. This one is called Candida albicans. You can see it's round, it's reproducing through budding here. And it happens to, Candida albicans is important from a clinical microbiology perspective because it does cause a lot of infections in the body. One of the most common infections that it causes is vaginal yeast infections, which is called vulvovaginal candidiasis. Right, so this is what happens when Candida albicans, which is normally located on the outside of the skin, gets into the vagina and causes itching and burning, for instance. That's called a yeast infection. Okay, so vulvovaginal candidiasis is given the name candidiasis because it's caused by Candida albicans, right? And it's abbreviated vulvovaginal candidiasis. And in order to get rid of it, you use a chemical called monostat. The monostat chemical interferes with the production of the yeast cell wall. This not the chitin, but it interferes with a chemical that you find in the plasma membrane of yeast and fungi called ergosterol. So that's a drug that kills yeast and fungi. It's the same. Athlete's foot is another disease, a fungal disease, a fungal mycosis, a disease that's caused by fungi. You actually kill athlete's foot with the same chemical that you use to kill vaginal yeast infections. Okay, so two examples of yeast, single-celled yeast that we've learned about are Saccharomyces cerevisiae and Candida albicans. Okay, let's look at fungi. As I said, the main difference between yeast and fungi is the fact that fungi can form multicellular colonies called mycelia, whereas yeast always exists as a single cell. Other than that, they're virtually identical. Okay, so here's a fungus called Penicillium chrysogenum. It's growing on an orange, right? So here we have Penicillium chrysogenum. And the name may sound familiar to you because the genus Penicillium is the fungus that was used to make penicillin, which is an antibiotic. So a researcher named Fleming, who was studying bacteria, pathogenic bacteria, discovered one day that a fungal colony was growing on one of his petri plates, and the bacteria were not growing where the fungal colony was. So he surmised that there must be, the fungus must be producing some chemical that's killing the bacteria. And after doing a lot of research, he isolated that compound, that chemical, and called it penicillin, and it was the first antibiotic. And as it happens, penicillin will not kill humans. It will not harm humans. It's basically harmless to humans, but it's deadly to bacteria, certain types of bacteria. So, fungi are multi-cell eukaryotes, but usually all of the cells are the same. Right? So there's a difference between a multicellular organism like a human, where the cells are all different because the cells have differentiated into different functions. There's a difference between a human kidney cell and a human heart cell. They look different, they do different things, they are turning different genes on and off, as opposed to a fungal colony where every cell is exactly the same. Right, so they're both examples of multicellular eukaryotes, but in humans the cells are differentiated to have specialized functions. That's true of all more advanced animals, whereas in fungal colonies all of the cells are the same. Okay, so they have a cell wall that's made of chitin, just like yeast. They are commonly called molds. The cells, when they form a multicellular colony, the cells form into little threads that are called hyphae. Right? So hyphae are little tiny threads that form the fungal colony, and each thread is made of individual cells. They reproduce by producing something called spores. Right? Spores can be dispersed easily in the wind, and so for the fungus that make you sick, you often get sick by inhaling these spores or having these spores float through the air and land on your skin. Now, are fungi haploid or diploid? Do they have one complete copy of the genome in each cell, or do they have two copies of the genome? Well, the answer is it depends on when you ask. There is a very brief period during the fungal life cycle where they are diploid. That's during the reproductive phase, but they spend most of their life cycle as haploid. They also have linear chromosomes, and if you catch a disease from having a fungus, we call those diseases mycoses. So a mycosis is a disease that's caused by a fungus. Collectively, we refer to all of the diseases that are caused by fungi as mycoses. So athlete's foot is a mycosis. A vulvovaginal candidiasis is a mycosis. Together, they are both examples of mycoses. Okay, so in this picture, you can see the fungal hyphae, these little threads that are made that are made of individual cells. Together they form a colony called a mycelium. All right, as I mentioned earlier, there was a scientist named Fleming who in the 1920s was studying a bacteria called Staphylococcus aureus that causes all kinds of bad infections. And so he was growing Staphylococcus aureus on Petri plates, and one day a fungal spore drifted in the window and formed a colony on one of his Petri plates, and he noticed that there was a clearance zone, a zone where there was no bacteria. That clearance zone is caused by the fact that this fungus is producing a chemical that is inhibiting the growth of the Staphylococcus aureus. It's preventing the Staphylococcus aureus from growing. So after many years, he isolated the active ingredient and called it penicillin. And penicillin was the first antibiotic. Penicillin doesn't hurt humans, but it kills specific types of bacteria. Okay, here's another type of fungus called Histoplasma capsulatum. Unlike Penicillium chrysogenum, Histoplasma capsulatum has no redeeming qualities. There's nothing good about it. And if you inhale the spores, you will get a very bad disease called histoplasmosis, which is where the spores sprout or germinate inside your lungs, and then you have fungus growing in your lungs. Some of those, one of the characteristics of fungi is that they tend to form colonies at low temperatures. A fungal colony, once again, a fungal colony is called a mycelium. And at low temperatures, anywhere from 16 degrees to 25 degrees, they will form a colony, a mycelium. At higher temperatures, fungi have the unique characteristic that they disperse into individual cells at higher temperatures, right? So fungi will form a colony at room temperature, but if they get into the blood where the temperature is 37, they will exist not as colonies, but as individual cells. They will look just like yeast because they're individual cells. Right, so geneticists have a word for that behavior. They say that fungi are dimorphic for temperature. Dimorphic, di means two, and morphic means forms. So dimorphic means something that exists in two different forms. So fungi are considered to be dimorphic for temperature because they exist as colonies at low temperature and they exist as individual cells at high temperature. So if you go on in biology, you'll hear the word dimorphic a lot, especially if you do genetics. You know, if a certain animal has two forms, one color versus another color, you say that it's dimorphic for that color. Most fungi are dimorphic for temperature. They exist as a colony at lower temperatures and they exist as individual cells at higher temperatures. Okay, so histoplasmosis, you inhale the fungal spores and you have fungus growing in your lungs where the temperature is relatively low, but in the blood you have individual fungal cells floating around in the blood and therefore dispersing itself to other parts of the body, and so it causes an infection that spreads throughout your entire system, throughout your entire body. When you have that kind of an infection, you call that a systemic infection, a systemic infection. If the infection is confined to just one small area, you call that, what would you guess? Right? You call that a localized infection. Right? So athlete's foot is an example of a localized infection because the fungus only infects the part of your body that stepped on the fungal spores. If you step on the floor of a locker room that has fungal spores on it, your feet will be infected with athlete's foot. That's a localized infection. If you inhale Histoplasma capsulatum spores, it will get into your lungs and then into your blood, and your whole body will be infected. That is called a systemic infection. Now, normally, if your immune system is working properly, if you're strong and healthy and have a strong immune system, your immune system can kill the fungus before it infects your whole body. If you're weakened because you've been sick or because you're undergoing cancer therapy or something like that, it can take hold, the fungus can take hold inside your body and cause all kinds of problems, and therefore we call it an opportunistic infection. Right? So any microbe, that anytime you have an infection from a microbe, but you're strong and healthy and you overcome it and it's not a problem, and it's only a problem when you're sick or you have a compromised immune system, like if you have HIV, AIDS, or if you're undergoing treatment for cancer, or you have anemia or other things. Under those circumstances, the microbe can get in and take over. We call that an opportunistic infection. An opportunistic means you take advantage of your opponent being weak, right? Opportunistically, you take advantage of a situation, even though it's kind of immoral and not honorable. That means that's what opportunistic means. Somebody's an opportunist if they, they exploit other people. If they exploit other people's weaknesses, they're called an opportunist. Right, so we have opportunistic infections. Right, so opportunistic infections are infections where your body is damaged by some microbe that would not normally damage your body if you were healthy. Okay. Okay, here is yet another fungus. This one is called Aspergillus flavus. Right, here it is growing on some corn. Right, and you notice that it's kind of a green, a brownish green color called khaki. Khaki is the color of army uniforms. Those clothes that soldiers wear are khaki green. So Aspergillus flavus is a fungus that has a khaki green color, and it is a fungus that's particularly bad because it produces toxins that are called aflatoxins. Aflatoxins are toxins that are not toxic until they pass through the human liver, and once they pass through the human liver, they are converted by the human liver into a poison, and they actually have the effect of causing liver cancer. So that's why it's always a bad idea to eat food that is contaminated with fungus. Sometimes you think, well, I'll just wash the fungus off, or I'll scrub the fungus, and I'll be all right. I can eat the food if I wash the fungus off. Well, that's not actually true, because the fungi, they send hyphae deep into the food, which you can't see. Right, so they're kind of, in this case, the hyphae are kind of like roots that grow deep underground. So you can cut the plant off the top, but you still have the roots underneath. So it's always a bad idea to eat food that's contaminated with fungus. You have to throw it out. Many people get liver cancer from eating food that's contaminated with fungus, but they don't know it right away because it often takes many years for the cancer to develop. So somebody gets liver cancer, they don't make the connection that they got liver cancer today because they ate a piece of moldy bread 10 years ago, but that's what happened. Okay, so speaking of fungi, remember that fungi produce spores, and many of them also produce aflatoxins. And if you inhale the spores, you can get a systemic infection. Even if you don't get a systemic infection, you may have an allergic reaction. You may have an anaphylaxis, an anaphylactic shock. So fungi are quite bad. They're quite a nuisance, really. And one of the main reasons that they're such a nuisance is that they produce these spores. The spores are the way that they disperse themselves over the environment. And spores are so small that they are easily carried by air, by wind. Right? So whenever you have fungal spores, it's dangerous. It's dangerous to be in an area where there are a lot of fungal spores. Now, most fungi can digest cellulose. Okay, so here we have a wall. Here's a wall. That's a building material that some of you may know. That building material is called drywall or gyprock. Gyprock is plaster with paper on either side. Right? And the purpose of drywall or gyprock, the purpose is to absorb moisture in your house to keep your house dry on the inside. So the drywall is made to absorb a little bit of moisture and keep your rooms nice and dry. But if you have a flood and you have a lot of stagnant water, stagnant means to sit still in the same place. Standing water is called stagnant water. If you have stagnant water in your basement or something like that, for a certain period of time, the gyprock will become wet. It will become wet enough that fungus can start digesting the cellulose that the paper is made of, that is on either side of the gyprock. So fungus will grow on wet gyprock. Fungus will grow on wet drywall quite easily. And so if you have flooding, it's common to have this kind of mold growing after the flooding recedes. Right? So if the inside of your house gets very wet, you'll get fungal growth all over the walls. And if you hire somebody to tear those walls out and replace them with new walls, they have to be dressed like this. So you notice that this builder is wearing goggles to prevent fungal spores from getting in his eyes, and he's wearing a respirator to prevent him from inhaling fungi, and he's wearing He's wearing a plastic suit to prevent fungal spores from getting all over his clothes, and then so that he inhales them later on. So you have to be very careful with fungus. Fungus is very bad for you. So if you have mold growing in your house due to dampness, it's very difficult to get rid of it safely. You have to be dressed properly. You have to hire the right kind of people that know what they're doing. Those people tend to charge a lot of money. So the truth is that if you have very, very extensive mold in a house, it's often better to just tear the house down and throw the components away and start again, because you can never really get rid of all the fungus. It'll come back, and it's bad for you. So because you can never really get rid of it, you may still have fungus growing in between the drywall and the wood that forms the outside of your house. It's better to, you can try to get rid of it, but that's very expensive, and it only costs a little bit more to tear the whole house down and start again. All right, so 3 examples of fungus that we discussed: Penicillium chrysogenum, Histoplasma capsulatum, and Aspergillus flavus. Those are the 3 examples of fungi that we learned about right now. Let's talk about bacteria. Okay, so here is a bacteria. Do you remember what the morphology of the bacterial cells are? They are longer than they are wide, right? And when you have something that's longer than it is wide, you call it a bacillus. Right. This is Lactobacillus acidophilus because, as I mentioned in the first lecture, they eat lactic acid. They like to absorb lactic acid, and they convert it. They eat lactose, which is a it's a polysaccharide. It's a disaccharide found in milk, and you also find it in some other areas of the body. That's one of the types of polysaccharides it likes to eat, but it's also perfectly happy to eat other things. But when it eats lactose, it converts it into lactic acid and then puts that out into the environment where it's living, and then that lowers the pH of the environment. But that's okay, because Lactobacillus acidophilus is an acidophile. It's an acidophile. So an acidophile is an organism that likes to live in an acidic environment with a low pH. So Lactobacillus acidophilus is a bacillus bacteria. It's a haploid. It ferments lactic acid and turns it in. It ferments lactose, the sugar, and converts it into lactic acid. Okay, so bacteria are small single-celled prokaryotes. They don't exist in colonies. They kind of grow together in clusters, but they're not physically attached to each other. So you don't call a colony of bacteria, you don't call that a multicellular organism, the way you call a fungal colony a multicellular organism, for instance. Okay, so bacteria are small single-celled prokaryotes. They're much smaller, on the order of 1 micron. They reproduce through binary fission. There is a strange form of sexual reproduction among bacteria that's called conjugation. We'll talk about that later. But most of the time, bacteria simply reproduce through binary fission. It means they split in two. They split in two equally, and they form 2 daughter cells that are of equal sizes. They are haploid. They have only 1 copy of the genome. They have a DNA genome, never an RNA genome. They have a single chromosome. Usually that single chromosome is circular. They sometimes have extra chromosomes or accessory chromosomes that are small, round chromosomes that are called plasmids. Plasmids are small extra chromosomes that you find in bacteria. Genetic engineers exploited this when they discovered it. They said, hey, we can extract these small chromosomes quite easily from bacteria, and we can put foreign genes into them, and then we can put them back in the bacteria and get the bacteria to make recombinant proteins. So in the first lecture I mentioned insulin. Insulin these days is produced in bacteria, not in the pancreases of pigs. Some bacteria have a cell wall that is made of a polysaccharide called peptidoglycan. Now, we'll learn very quickly in the next lecture, we'll learn that there are 2 types of bacteria, those that have a cell wall and those that don't. Those that have a cell wall are referred to as gram-positive cells because they react with a certain stain that's called the gram staining protocol. So a bacteria that has a peptidoglycan cell wall is classified as a gram-positive bacteria. A bacteria that does not have a cell wall is classified as a gram-negative bacteria. So that all of the bacteria in the world are classified according to one of 2 categories, either gram-positive or gram-negative. That classification is based on whether they have a cell wall or not. Here's an interesting thing. Lactobacillus acidophilus is a normal part of the human intestine as well as a normal part of the vaginal environment. Right? So Lactobacillus acidophilus lives in the human vagina, right? And it is part of what's known as the normal flora or the normal microbiome of the vagina. So it's found in the vaginas of healthy women. It's normal. Not only is it normal, but it's a good thing because lactobacillus living in the vagina produces lactic acid, which lowers the pH of the vagina. So the pH of the vagina is lower, has a lower pH than the rest of the body, and that prevents Candida albicans from getting into the vagina because Candida albicans is not an acidophile. Candida albicans doesn't like a low pH. Right? So that is why the times that a woman gets a yeast infection, it's usually caused by one of two things. Number one is washing the vagina too much. If you wash the vagina too much with a douche or something, that washes away all of the Lactobacillus acidophilus, and then the Candida albicans can come inside from where the skin where it lives on the outside. On the other hand, if a woman is sick with some kind of a bacterial infection and they give her antibiotics to kill the bacteria that's infecting her, those antibiotics will also kill the Lactobacillus acidophilus that's in the vagina, and then that will cause her to get a yeast infection. That gives the Candida albicans on the skin the opportunity to come inside and cause a yeast infection. So again, that is called an opportunistic infection. The Candida albicans is taking advantage of the opportunity to get into the vagina while the Lactobacillus acidophilus is gone. Right? So Candida albicans is the yeast that causes yeast infection in the human vagina. Lactobacillus acidophilus is the bacteria, the good bacteria that's usually there that prevents the Candida albicans from getting in because it creates an environment that Candida albicans doesn't like to live in. So interestingly, humans, we are covered on the outside by billions of bacteria and billions of different types of yeast and a few fungi. The inside of our digestive system is filled, the intestine in particular, is filled with different types of bacteria and yeast, as well as the vagina is filled with lots of different bacteria, specifically Lactobacillus acidophilus. And all of these bacteria are living in an ecosystem. You learned about ecosystems in Biology 110, that an ecosystem is where you have all these different plants and animals that interact with one another and they maintain a balance. The predators eat a certain amount of the prey eat a certain amount of the plants. That prevents the plants from overgrowing. When the parts of the plants die, the detritivores recycle the materials into other macromolecules. That's all called an ecosystem. And believe it or not, the human body is an ecosystem as well that's populated by billions of different bacteria, fungi, and so on, and they all live in a balance in a healthy person. Sometimes the different bacteria and different bacteria and yeast that live in the human intestine get out of balance, and there's too many of these and not enough of those, and that causes you to be in poor health. And that's why they encourage us to take something called probiotics. Right? So antibiotics are drugs that kill bacteria. Probiotics are pills that are filled with freeze-dried bacteria like Lactobacillus acidophilus, and you take that and you put the good bacteria back into your intestine and restore the balance so that you can be a healthy person again. So it's not particularly healthy to have no bacteria in the intestine. It's healthy to have the right mixture of bacteria in your intestine. Also the right mixture of bacteria in the vagina, for instance. And so remember that Lactobacillus acidophilus is the bacteria that prevents Candida albicans from causing vulvovaginal candidiasis. Okay, we mentioned the fact that bacteria can either be rods or cocci. Rods are the technical term for a rod is a bacterial cell that's longer than it is wide. The technical term for a rod is a bacillus. Right? And so rods or bacilli are bacteria that are longer than they are wide. If they are round, if the bacteria are round instead, we call them a coccus, or plural cocci. And notice that, so these bacteria are called Staphylococcus aureus. Right? Staphylococcus. So again, the shape of the bacteria is given to you in the name. Right? It's given to you in the name. So you can tell just by reading the name that this bacteria will be round. It's a coccus. Okay, now furthermore, The word staphylo means in a cluster. So notice how these round bacteria are clustered together like that. Right? So anytime you hear the word staphylococcus, you can expect to see round bacteria forming in clusters. Right? What would you expect to see if you heard the word staphylobacillus? You'd expect to see rod-shaped bacteria in clusters. Okay, now there's another word that we're going to learn later called strepto. So streptococcus, the word strepto means in a chain. Right, so a streptococcus, like we're going to learn about a bacteria called Streptococcus pyogenes. Streptococcus is a bunch of round bacteria in a chain. Right, so these are in a cluster, so these must be staphylococci. And this is a particular type of staphylococcus called genus Staphylococcus, species aureus, Staphylococcus aureus, which is a common. It is commonly found. It is part of the normal flora or part of the normal skin microbiome. The microbiome is the ecosystem that you normally find on the skin or the intestine or something. So I mentioned earlier that the gut microbiome is the normal ecosystem of bacteria and fungi and yeast that you find inside the intestine. Okay, so the skin microbiome includes Staphylococcus aureus, Candida albicans, Saccharomyces cerevisiae, a few other types of bacteria. Right? And so that they are all part of the normal skin microbiome, or what is also called the normal flora of the skin. The word flora is a more primitive word. Microbiologists in the early days of microbiology, they used to refer to ecosystems of bacteria as flora. You remember that the term fauna from Biology 110, the word fauna means animals, the normal animals that you would find, and then flora means the plants that you would find. So people were kind of foolishly calling bacteria plants by referring to them as the normal flora. It's a misnomer. A misnomer is a word that's badly chosen, or a word that is kind of confusing in its meaning. But it is used. This term is used. So the term normal flora, you'll see a lot of older bacteriologists like me sometimes referring to ecosystems of bacteria as the normal flora of the skin or the normal flora of the gut. The modern term, the correct term, is the gut microbiome or the skin microbiome. Okay, now Staphylococcus aureus is normally harmless because it stays on the outside of the skin. As long as it stays on the outside of the skin, it's harmless. If you have a wound so that it can get inside the skin, that becomes an opportunistic infection. Right, so if you have a wound and Staphylococcus aureus infects it, we mentioned earlier that Staphylococcus aureus is a pyogenic bacteria. So if you have a wound that gets infected with Staphylococcus that is normally found on the skin, that wound will develop pus. It will develop that sticky yellow stuff that smells bad called pus, and what pus is a whole bunch of dead white blood cells, a whole bunch of dead macrophages that have shown up in that area to eat the Staphylococcus aureus. Okay, another common infection, when you go to the hospital, sometimes if you're very sick and you can't urinate on your own, they will put in something called a catheter. It's a kind of a painful procedure where they put a tube inside the urethra and up into the bladder, and therefore the urine can come out without your having to urinate. Now, if that plastic tube that they put into your urethra is dirty with Staphylococcus aureus, you'll get a bladder infection. So that's called a urinary tract infection, or a UTI, urinary tract infection. And the most common urinary tract infections are caused by people putting in dirty catheters where you accidentally touch the catheter to the skin and then you push it in through the urethra. Right? And so the catheter will pick up some Staphylococcus aureus from the skin and carry it up the urethra into the bladder, and then you have a urinary tract infection. We'll talk about this later, but the epidemiologists have a word for that. They call it an iatrogenic infection. An iatrogenic infection is an infection that you get as a result of a specific medical procedure. So if you go to the hospital and they do a specific thing to you, and then whatever they did to you results in you're getting some kind of an infection, they refer to that as an iatrogenic infection, and we'll talk about that in detail later. Okay, another interesting thing that Staphylococcus aureus can do is cause food poisoning, which is called staphylococcal food poisoning. Under the right conditions, most bacteria will double. They will divide through binary fission. They will double every 20 minutes at 37 degrees or thereabouts. Now, what bacteria like to eat are sugars and to a certain extent proteins. Okay, so this pie that I've shown you up here at the top is called a custard pie, which is made entirely of sugar and egg white. Right? You may have had a custard pie. Right? That's a very rich environment for bacteria. Right? So, if somebody cuts that pie, let's say somebody takes that pie to a picnic on a sunny day. So you're outside with the family in the hot sun. It's maybe 40 degrees outside. Somebody touches that pie and infects it with Staphylococcus aureus from their skin. Let's say they deposit a couple of hundred Staphylococcal aureus cells onto that pie by touching it. That would be very common. The Staphylococcus aureus is already on your skin. You transfer 200 cells to the surface of that pie. You leave that pie out in the sun for 8 hours, and that bacteria, those 200 cells, will double every 20 minutes. Right? So you've got 200, then you've got 400, then 1800, 1600, 3200. After about 8 hours, you will actually have billions of Staph aureus cells growing on that pie, and if somebody eats it, they'll get staphylococcal food poisoning. Right? So staphylococcal food poisoning is caused by people touching food, getting Staphylococcus aureus from their fingers onto the food and then leaving it at a high temperature so that the Staph aureus can grow, and then when you eat it you become sick with staphylococcal food poisoning. Okay, as I said, the word strepto means a strip or a chain. So here's another common bacterium called Streptococcus pyogenes. This is also part of the normal skin microbiome, or part of the normal flora of the skin. Right, so you can see that they're forming chains here. Streptococcus pyogenes is already on the skin, but occasionally in children, for instance, some of it gets inside their mouth or down their throat or up their nose, and it causes a systemic infection called scarlet fever. Scarlet fever can cause an infection of the heart called bacterial endocarditis, which causes damage to the heart. And so if you catch scarlet fever as a child, you should get rid of the scarlet fever as quickly as possible with antibiotics. So if you're a mother and you see your child come down with this they develop a cough and they turn red like this and they develop a fever, take them to the hospital right away to make sure that they don't have a Streptococcus pyogenes infection. Because if that infection persists, you will get bacterial endocarditis, which damages your heart, and then you end up having heart problems in middle age. Right? So, in the old days, a lot of people used to die from scarlet fever because it damages the heart. But these days, if it's diagnosed quickly and then the Streptococcus pyogenes is killed quickly by antibiotics, then you don't have to worry about it. But you have to be on top of it. You have to pay attention. And so if your child comes down with a sickness like that, a fever and a red color like that, red tongue, red throat, red tongue, and a fever, take them to the hospital or the doctor immediately to check and make sure they don't have Streptococcus pyogenes. Now here's another interesting bacteria called Clostridium botulinum. You might have heard of food poisoning called botulism, right? Botulism is a type of potentially deadly food poisoning that is caused by this bacteria called Clostridium botulinum. It causes botulism. Now, botulism is what happens is if your food gets contaminated by Clostridium botulinum. Clostridium botulinum is an example of something called an obligate anaerobe. An anaerobe is a bacteria that doesn't like to live in air. It likes to live without air. So what happens is this bacterium makes little seeds that are referred to as endospores. The word endo means inside, of course, and then a spore is sort of like a bacterial spore. It's like a little seed. Bacteria are haploid, of course, so a seed is something that a plant makes, and a seed is always diploid. But this is sort of like a seed, except that it contains a haploid genome, not a diploid genome. So there's this little container called an endospore that Clostridium botulinum will produce, and this is its way of reproduction. This is its way of scattering seeds. And so if you make food, particularly canned food, and you got some Clostridium botulinum normally lives in the dirt. It lives in the soil. And if you get some dirt into the food and then you cook the food, you will kill the Clostridium botulinum, but you can't kill the endospores because the endospores are very difficult to destroy. And so the endospores get into the food when you can it. Let's say you make some jam, or you can some pears, you have some canned fruit that got dirty from soil. You cook the fruit so you think it should be safe, and then you put it in a can. The endospores will then germinate. The endospores will sprout, and then producing new Clostridium botulinum cells, and then those cells will start to eat the fruit, and they produce a gas. When they eat the sugars in the fruit, they produce gas, which causes the can to swell up. So whenever you see a swollen can like this, never eat what's inside it. Never eat the contents of a can like this, because it's contaminated with Clostridium botulinum. Clostridium botulinum is producing a toxin that causes paralysis of the muscles. Right? That toxin is called, can you guess? It's called Botox, because it's produced by Clostridium botulinum. And Botox has the effect of paralyzing muscles. Right? So one of the most important muscles you need to live is the diaphragm, which you use to breathe. And so if somebody catches botulinum from eating contaminated food like this, they could potentially die because they lose the ability to breathe. So initially they'll have trouble breathing. They'll feel numb and have trouble breathing, and then they get taken to the hospital, and then they'll be intubated, meaning they put a tube down your throat and attach it to a ventilator so that you can breathe. And so that's what they would do if they caught you, if that was diagnosed. But if you were away someplace, all by yourself, it could easily kill you, because you don't have a hospital or a ventilator or somebody to put the tube down your throat. There's nobody there to intubate you. Okay, so Clostridium botulinum is an anaerobe. It lives in the soil. It doesn't like the air. It likes to live without air. It produces endospores, and those endospores can survive cooking. Right? So the endospore, even if you cook the food, if it has Clostridium botulinum endospores, once you put the food into a can or into a jar, those endospores will germinate and produce new Clostridium botulinum cells that go to work eating the food, producing a gas, causing the can to swell up. That's a telltale sign that you shouldn't eat what's inside there. As I said, the toxin that is produced by Clostridium botulinum is commonly called Botox. What it does is it causes paralysis of muscles. It causes something called flaccid paralysis. Flaccid means limp, right? And paralysis. So basically, Botox causes your muscles to go limp, so that you can't contract your muscles. Now, why do you use Botox? Why do you use Botox in cosmetic surgery? If you inject small amounts of Botox into your forehead, it prevents the muscles of your forehead from furrowing your brow, right? So it prevents you from raising your eyebrows, which causes wrinkling. And so if you basically paralyze the muscles above your eyebrows, then you can't furrow your brow. You can't make creases in your forehead. Right? So that's why Botox is used as a treatment in cosmetic surgery. Eventually, do you put enough in? Do they inject enough to make you die from flaccid paralysis because you can't breathe? Obviously not. They put in just little amounts. They inject small amounts. Those small amounts will eventually disperse and travel throughout the body. There's not enough of it to do any real harm, and then they have to go and inject some more. But that toxin that causes paralysis of the muscles in your forehead, so you won't get a wrinkly forehead, is called Botox, and it's originally produced by Clostridium botulinum, and that is what kills people when they get botulism. Okay, so 4 bacteria that we need to know: Lactobacillus acidophilus, Staphylococcus aureus, Clostridium botulinum, and Streptococcus pyogenes. Those are 4 bacteria that we need to learn the names of as soon as possible, because I'll keep asking you questions about them on all the quizzes. So remember all of these names as soon as you can, because I will be asking you about them. Because I've taught you about these organisms now, I'll be asking about them in future quizzes. Okay, so memorize these 4 types of Clostridia as soon as you can. Right? So there are actually 4 members of the genus Clostridium. There is Clostridium botulinum, which causes bacterial food poisoning, botulism, and it causes the effect that the Botox causes the effect of flaccid paralysis. Flaccid paralysis means your muscles go limp and you can't breathe because you can't contract your diaphragm. Okay, there's a closely related cousin to Clostridium botulinum called Clostridium tetani, which causes a disease, an illness called tetanus. And this is what happens if you cut yourself with a dirty knife. The Clostridium tetani gets into your blood and produces the Clostridium tetanus nerve toxin, which causes something called rigid paralysis. Rigid means stiff, of course, and inflexible. So what happens is this neurotoxin is very similar to Botox, except that it causes your muscles to become rigid and stiff instead of flaccid and limp. Right? So again, you can't breathe because you flex your diaphragm once and it never relaxes again, so you can't breathe. So tetanus kills people as well by preventing them from being able to breathe. All right, there's another cousin to Clostridium botulinum called Clostridium difficile, C. difficile, which is a common cause of something called hospital-acquired diarrhea or antibiotic-associated diarrhea, which is a disease you get in the hospital. What happens is, if you go to the hospital because you have a bacterial infection of some sort, and they give you a course of antibiotics to kill the bacteria that's infecting you, those antibiotics will also kill the bacteria that are living in your intestine. Lactobacillus, Escherichia coli, a lot of those other things. And that gives Clostridium difficile the opportunity to get into your intestine while it's unoccupied, right? So this is an opportunistic infection. It's an vulvovaginal candidiasis, which I just talked about, is an opportunistic infection. So is this. In this case, we have used the antibiotics have killed off the good bacteria that are in our gut microbiome. And this gives the opportunity for some bad bacteria to go in there while it's unoccupied. And if Clostridium difficile gets in there, then you end up getting hospital-associated diarrhea. So that is a C. difficile outbreak in a hospital is a very common thing. Very often you'll see hospitals once in a while, not often, but hopefully not often, but once in a while you'll see hospitals having an outbreak of C. difficile infection. Okay, finally, the 4th member of the Clostridium genus that we have to worry about is called Clostridium perfringens, which causes gangrene, or more accurately, gas gangrene. So gangrene is where you get a wound. Usually you cut yourself, the wound turns black, and then that blackness, necrosis, spreads up and down your arm, and there's no way to stop it unless they cut your arm off. They used to amputate limbs when people got cuts and wounds and things like that. They used to cut off your arm or your leg rather than risk having you come down with gangrene. Right? So gangrene is caused by another member of the Clostridium genus called Clostridium perfringens, and all 4 of these bacteria live in the soil, and therefore the source of all 4 of these bacteria getting into you is through dirt. Right? So your food is contaminated by dirt. In the case of Clostridium botulinum, your food is often contaminated with some dirt that contains Clostridium difficile as well, but it never has a chance to catch hold in your intestine. It never has a chance to colonize your intestine because there are already a whole bunch of bacteria living there that do not harm you. And then Clostridium perfringens and Clostridium tetani usually get into your body through cuts using dirty implements. So you cut yourself with a sharp knife that was dirty with soil, you could get tetanus, or you could get gangrene. Viruses are not actually living things. They are a few genes contained in a little container called a capsid that go around infecting live cells and then hijacking the cell's metabolic machinery and using that metabolic machinery to make more viruses. That's what a virus is. Okay, so here's an example, influenza virus. As I said in the last lecture, they're very small, so you can see the size, this scale here, of 25 nanometers. Right, so this is this is probably 50 nanometers from one side to another. Remember that there are 1,000 nanometers in a micrometer, which is already quite small. Okay, so here we have a virus, influenza virus in this case, and inside we have 8 different chromosomes that are made of RNA. Right? So the chromosomes, these 8 different chromosomes contained inside this viral capsid, as it's called. Right? So the container that contains the genes is referred to as a capsid. We have inside the viral capsid, we have the viral chromosomes. In this case, they're made of RNA. So this thing gets into your cells, and it releases the genes, which are made of RNA. The RNA genes load themselves directly onto ribosomes, and the ribosomes translate them into proteins, thinking that they are, just regular messenger RNAs, and then you end up making more viruses. Okay, so viruses are not really forms of life. They tend to be, they're just a few genes contained in a capsid. They are very small, in the nanometer range. They contain only a few genes generally in the capsid. Some of them have one chromosome, right, and others have more. So influenza is an example of a virus that has several chromosomes. That causes some problems, as you'll see when we discuss the influenza virus. And there are some viruses that have DNA genomes, like the smallpox virus, for instance, has a DNA genome. The influenza virus has an RNA genome. So some viruses have an RNA genome, some have a DNA genome. There are examples of both types. Okay, so here is an actual electron microscope image of an influenza virus. It's so small that we need to use a special type of electron microscope image called a shadowing TEM effect. But they are quite small. So this is an actual image of an influenza virus. This is an image of another virus called the varicella-zoster virus. It's a DNA virus. It has a single linear chromosome. It's about 0.2 nanometers in diameter. It's quite small, and it is a teratogen. Right. So if you catch varicella-zoster when you're pregnant, the baby could have birth defects. Right. So avoiding catching varicella-zoster when you're pregnant. We usually have a vaccine that prevents you from getting varicella. So it's usually not a concern for most people. All right, here's another virus called the hepatitis A virus, which enters the body through something called the oral fecal route. Means that you eat it and then you transmit it to other people through your feces. In this case, most people catch, by the way, hepatitis, what organ do you think it affects? Right? The word hepato refers to which organ? Refers to the liver, of course. Right, so hepatitis A virus is one of 3 viruses that affects the human liver. We also have hepatitis B and hepatitis C, and you usually catch it by eating food that has been contaminated by small amounts of excrement on the fingers of the people who prepare your food. So that's why it's always a good, it's a requirement that people who prepare food do a very good job of washing their hands. And even that usually isn't good enough. Food preparation technicians, chefs and cooks and people who put together sandwiches should always be wearing gloves as well. Right? So that's a way of preventing the spread of hepatitis and other viruses that can easily get on your fingers. Okay, so 3 examples of viruses that we've talked about are the influenza virus, the varicella-zoster virus, and the hepatitis A virus. Okay, prions. Prions are actually small proteins that can reproduce themselves without the need for a gene in between. Example, mad cow disease was something that struck Europe in the 1980s, and the technical term for it is bovine spongiform encephalopathy. Right? Bovine refers to cow. Encephala refers to the brain. Right? So spongiform basically just means this particular protein causes the brain to start to break down, initially causing the cow to act funny. But this is a prion, and we're not really going to study prions in this course, but you should be aware of what a prion is. Right? So be aware of what a prion is. Okay, so this is a summary of the representative microbes that we have studied. Right, so the bacteria that we've studied include Lactobacillus acidophilus, Staphylococcus aureus, Clostridium botulinum, and Streptococcus pyogenes. You should try and memorize those as soon as you can, because I'll start asking multiple choice questions about those. The fungi that we've learned about are Penicillium chrysogenum, Histoplasma capsulatum, and Aspergillus flavus. The protozoa that we've learned about are Plasmodium vivax, Giardia lamblia, and Toxoplasma gondii. The yeasts that we've learned about are Saccharomyces cerevisiae and Candida albicans. The viruses that we learned about are varicella zoster and hepatitis A virus, also influenza virus, but we won't worry about that yet. And the helminths that we learned about are Diphyllobothrium latum, which is a tapeworm, and Trichinella spiralis, which is a nematode worm. We'll learn what the difference between those two is later on when we study the helminths. Right, so these are all the different representative microbes that we've learned about on our tour in this lecture. Now, why don't we just do some relatively easy practice questions? These are the sorts of questions that I will start asking you probably even on the first quiz. Right, so let's see how well we can remember some of these things. Which of the following are eukaryotes? You can stop the recording and see if you can answer that. The answer is the helminths, the protozoa, humans, fungi, and yeast are all examples of eukaryotes because they all have a nucleus that contains the genetic information. Okay, which of the following of these are not actually living things? And there's more than one on the list here. And the answer is viruses and prions are not alive. Now, viruses are just a couple of genes inside a container called a capsid, and prions are proteins that contain no genetic material at all. Which of the following have circular chromosomes? The answer is bacteria and some viruses. Pretty much all bacteria have circular chromosomes, and they only have one chromosome. They also have a few additional chromosomes called plasmids. Some viruses have circular chromosomes, and some viruses have linear chromosomes. Which of the following have an RNA genome? Well, based on the question I just asked, you should be able to answer that one fairly easily. Right? The answer is some viruses, like the influenza virus or like the coronavirus that's causing the pandemic that we're in right now, some viruses have an RNA genome. Other viruses have a DNA genome. Okay, which of the following are prokaryotes? Okay, it's a little bit of a trick question because there's only one. Right? Bacteria are the only examples of prokaryotes. All bacteria are prokaryotes. All prokaryotes are bacteria. Which of these are made of more than one cell? Okay, the answer is the helminths, the humans, and the fungi. Fungi, do they consist of, are they multicellular? It's sort of on the border. It's sort of a yes or no. Some people would classify them as multicellular, some would not. Definitely the helminths and humans, of course, are multicellular organisms. Helminths are full-fledged multicellular organisms with fully differentiated cells. So they have different cells that are specialized to do different things, the same way that humans do. Okay. Candida albicans is an example of which type of organism? Candida albicans, of course, is a yeast. Why can't it be a fungus? Can we call it a fungus? It is sort of true that yeasts are fungi. They're sort of classified as fungi, but the difference between a yeast and a fungus per se is that a yeast is never multicellular. It never forms a colony. It usually doesn't. It doesn't form a colony. It usually exists as a single cell. Okay, Plasmodium vivax is an example of which type of organism? The answer is it's a protozoa. It's a protist. Protist is the correct term. Protozoa is a specialty term. Okay, lactobacillus, what is it? Answer, it's a bacteria. Penicillium chrysogenum, what is it? Answer, it's a fungus. It's a fungi. Diphyllobothrium latum, what is it? Answer, it's a helminth. It's a tapeworm. Okay, arrange the following organisms in order from the smallest to the largest. Okay, you might want to stop the recording and see if you can do that. Okay, I'll answer. Okay, so the answer would be, which one is the smallest? Okay, now you have two that are possibly the smallest. We have the viruses and the prions. Generally, a protein is smaller than a virus. So the smallest is the prion. The second smallest is the virus. Okay, which one is the largest? That's easy. Okay, the largest, of course, is the human. Okay, now, which one, next to a virus, which one of these is smallest? Okay, the next thing up from a virus would be a bacterium. And then after a bacterium, which one would be larger? Yeast cells and fungal cells are about the same size, and so they would be next. Okay, then protozoa. Protozoa are large single-celled eukaryotes, right? Fungi and yeast are regular-sized single-celled eukaryotes. Protozoa are large single-celled eukaryotes. They tend to be 50 to 100 microns or even bigger sometimes. And then, of course, the helminths are bigger still because they're multicellular eukaryotes, and in some cases they can grow to become enormous. 10-meter long tapeworms are enormous. All right. You will have to memorize some genus and species names, so let's see if you can do any of these. All right, starting with the first question up at the top there. What is the main difference between a prokaryote and a eukaryote? What would you say the main structural difference is? Right. This is an answer that I would give. The main difference is that that a eukaryote has a nucleus and a prokaryote doesn't. But a eukaryote is also bigger than a prokaryote, generally speaking. Okay, I gave you the answer too soon. I asked, give the genus and the species name for one common skin bacterium that causes staphylococcal food poisoning. The answer would be Staphylococcus aureus. The other question down here, the other question up at the top is a helminth a prokaryote or a eukaryote? What is the answer? Helminth is a multicellular eukaryote. Okay, then this other question down here at the bottom, give the genus and species name for the bacterium that causes scarlet fever in children. Do you remember what it is? And the answer is Streptococcus pyogenes. Okay. Top question, give the genus and species name for the microscopic roundworm that causes trichinosis when you eat undercooked meat. What's the answer? Trichinella spiralis. Give the name of the virus that damages the liver. Give the name of a virus that damages the liver and is commonly transmitted via the fecal-oral route by food handlers who don't wash their hands properly after using the toilet. That means they get a little bit of excrement on their hands after using the toilet. They don't wash it off properly, and then it gets into your food, and then you catch hepatitis A. Right? Hepatitis A is not particularly bad. Of the three, it's the mildest. Of the hepatitis A, B, and C, hepatitis A is the mildest. It usually doesn't damage you particularly, but why catch it if you don't have to? Right? What is the hepatitis A is the answer. Right? Okay, so give the genus and species name of the common skin yeast that causes yeast infections, also known as vulvovaginal candidiasis. Okay, so the name vulvovaginal candidiasis should be a dead giveaway. It should be an easy question. The answer is Candida albicans. Give the genus and species name for the common industrial yeast that we use to prepare beer and wine. What's the answer? The answer is Saccharomyces cerevisiae. Give the genus and species name for the protist that causes malaria. Do you remember it? Okay, so there are several different members of the same genus, but let's just give out one name, the answer being Plasmodium vivax. Plasmodium vivax causes the most severe form of malaria. Other ones cause lesser forms. All right, next question. Give the genus and species name for the common aerobic spore-forming soil bacterium that can cause botulism in foods that are improperly prepared or preserved. Okay, so the word botulism should be a giveaway. That should be a big clue as to what the answer is. What is the answer? The answer is Clostridium botulinum. Finally, give the genus and species name for a common fungus that is often it's a soil. It grows on it spoils grain crops like corn and peanuts, and it produces an aflatoxin that causes liver cancer. So do you remember the name of that khaki green fungus that we saw growing on the corn earlier in the slideshow? And the answer is Aspergillus flavus. All right. Take some time to go through this list and then tell me which of these are bacteria. Right, so if this was on a piece of paper, I would ask you to circle the ones that are bacteria, but you can tell me which ones are bacteria. You can stop the recording if you need time. All right, so these are the bacteria. This is the answer. Clostridium botulinum, Lactobacillus acidophilus, Staphylococcus aureus. Which of the following bacteria are commonly which of the following is a bacteria that is commonly found on the skin, but which can cause wound and catheter infections? Do you remember which one it was? Answer is Staph aureus, Staphylococcus aureus. Hey, which of the following is a spore-forming bacteria that causes a deadly form of food poisoning? I'm not telling you what the food poisoning is called because that would make it way too easy. What's the answer? Clostridium botulinum. Which of the following is a spore-forming bacteria that produces a toxin commonly used in cosmetic surgery? You inject it to stop your forehead from wrinkling. The answer is Clostridium botulinum again. Which of the following is a bacteria that can prevent vaginal yeast infections? The answer is Lactobacillus acidophilus. Which of the following are yeasts? The answer is Candida albicans and Saccharomyces cerevisiae. Which of the following is the cause of common vaginal yeast infections? So the Lactobacillus acidophilus is excluding one of these yeasts and therefore preventing vulvovaginal candidiasis. Which fungus is it? Obviously, Candida albicans. Which of the following is the cause of vulvovaginal candidiasis? That gives the name away right there. The answer is Candida albicans. Which of the following yeast is found on the skin and elsewhere, but is commonly used for industrial brewing, making wine, beer, and bread? It is also known as baker's yeast or brewer's yeast. Which one is it? The answer, of course, Saccharomyces cerevisiae. Which of the following are fungi? The answer, Histoplasma capsulatum, is the only fungus on the list. Okay, which of the following cause histoplasmosis? That should be an easy one. The answer is Histoplasma capsulatum. Which of the following are protozoa? Okay, take a minute to find the protozoa, and the answer is Giardia lamblia, Plasmodium vivax, and Toxoplasma gondii. Which of the following is found in contaminated drinking water and causes traveler's diarrhea? And the answer is Giardia lamblia. We have Giardia lamblia here in British Columbia, and people who go on camping trips do catch giardiasis. Which of the following causes giardiasis? And the answer, of course, Giardia lamblia. It's in the name. Which of the following can be found in cat feces and can lead to birth defects if it infects a pregnant woman? Which one is it? The answer, Toxoplasma gondii. Which of the following causes malaria? The answer, Plasmodium vivax. Which of the following are helminths? The answer, Diphyllobothrium latum is the only one on the list, and we learned about Trichinella spiralis as the other one, but it's not on this list. Which of the following is a parasite contracted from eating undercooked fish? The answer, of course, the same thing, Diphyllobothrium latum. Which of the following causes diphyllobothriasis? Very easy at this point. The answer, Diphyllobothrium latum causes diphyllobothriasis. Diphyllobothriasis is the symptoms associated with having a gigantic tapeworm in your large intestine. Which of the following are teratogens that cause birth defects if pregnant women catch them? Varicella zoster and Toxoplasma gondii. Which of the following are viruses? Answer, Varicella zoster is the only one on the list. Which of the following causes a disease that's commonly referred to as chickenpox? Answer, Varicella zoster. Okay, now a little bit more challenging. Can you name the microbes that cause the following diseases? Malaria, giardiasis, and vulvovaginal candidiasis. Right, the last two should be easy, but what are the answers? The answers, okay, sorry, you know what the answers are, right? Candida albicans. I've even forgotten the question now. Okay, so what you've learned today, we learned the different branches of microbiology in the first slideshow, and we learned the general difference between prokaryotes and eukaryotes. We learned how to do binary nomenclature for naming. We learned, and then we have just now learned a few examples of each of the following types of organisms. We learned a few examples of bacteria, a few examples of protists or protozoa, a few examples of helminths, a few examples of fungi, a few examples of yeast, a few examples of viruses. And we had some sample exam questions. Those are the typical types of questions that I would ask on a quiz. All right, and now go on to lecture 3, where we'll talk briefly about the different kinds of microscopes that we use to look at bacteria and viruses and other microscopic organisms. And for a lot of you that will be a review, but you can take a look at that slideshow anyway.